दोस्तों कैसे हो? आप लोगों ने जो रिस्पॉन्स, अफेक्शन, रिस्पेक्ट दिया है, वी आर टच्ड। आप लोग अच्छा करें लाइफ में, बस वी वांट यू टू बी बेस्ट। ओके, इस वीडियो में एक कंप्लीट पेपर डिस्कशन दिया है CUET लाइफ साइंस 2021 का। हम उम्मीद करते हैं कि इट विल हेल्प यू अ लॉट एंड यू विल अगेन गिव लव टू दिस वीडियो एज वेल। चलो शुरू करते हैं। CUET PG लाइफ साइंस का जो पेपर होता था वो दो सेक्शन में आता है। 25 क्वेश्चन जो इनिशियल होते हैं वो आपके इंग्लिश लैंग्वेज, जनरल अवेयरनेस, मैथमेटिकल एप्टीट्यूड एंड एनालिटिकल स्किल्स के होते हैं। जरा सिंपल सा अगर आप ध्यान से देखेंगे तो आपके इसमें से काफी क्वेश्चन बन जाएंगे। सेक्शन बी जो होता है वो 75 क्वेश्चन होते हैं बायोलॉजिकल डोमेन के, जिसमें के आपकी एक्सपर्टीज है। तो शुरू करते हैं डिस्कशन फर्स्ट पार्ट ए का एंड देन फॉलोड बाय पार्ट बी का।
Select the correct option in the given statement. You may feel relieved when something bad happens, does not happen, is remembered, or occurs. As we know, feeling relieved means feeling good or relaxed as something difficult problem has passed through. So you start feeling relaxed when you are not worried about something bad. So here the option B would be more appropriate, that you may feel relieved when something bad does not happen. Select the most suitable synonyms of barbarian. It's impolite or unkind or uncivilized or unlikeliness. Synonyms of barbarian is savage, uncivilized, rough, hooligan or rude. So option C is more close to the barbarian term. Which is the closest meaning of the phrase to be hard of hearing? Deaf, deaf and dumb, careless or arrogant. Hard of hearing refers in part to a person who is having compromised hearing ability or deafness. For example, we often say that we have to be a bit louder as our grandparents is a little bit hard of hearing. So here the option A would be the correct answer. Identify the meaning of the idiom from the option given in black and white. Is it useless? Is it in short, or in writing, or in full swing? In black and white means in writing or print, which is more reliable than mere verbal commitments. For example, we often say, I won't believe verbal commitment until I see it in black and white. So option C is the correct answer. Select the most suitable antonym of the rapture. Is it despair, elation, ecstasy, or hell? Rapture basically represents the pleasurable emotions. The synonyms of rapture would be ecstasy, elation, euphoria, paradise, while depression or despair are the common antonym of rapture. So here the option A would be the correct answer. Complete the second sentence by selecting the appropriate options. One way to improve your health is to eat more fruit and vegetables alternatively. So it is very simple. As we know, following healthy diet and regular exercise, they are the key of prolonged life. So option C would be the correct answer, that one way to improve your health is to eat more fruits and vegetables. Alternatively, you could exercise more. Fill in the blanks with the right word. An employee might be given a privilege as a form of punishment, increment, reward, or commendations. Privilege, they are often given as a reward to promote or encourage an individual at the workplace. Unlike increment, privilege are the temporary and sometimes non-monetary as well. So option B would be the correct answer. Select the correct option for the use of the word the Delhi, the city of Delhi, the southern India, or the Long Island. The word is often used in front of unique, definite, or specific nouns. So option C and D cannot be the answers. Why? Because they are not the specific or definite nouns. Now option A and B could be the answers. But if you see in option A, Delhi is a proper noun. And generally we do not add the word the in front of a proper noun. So option B would be the correct answer. Fill in the blank with the correct propositions. I am worried dash exam. On, in, about, or of. A preposition is a word which comes before a noun or a pronoun and establish a relationship between elements of a clause or a word. Option C would be more close to correct because it is connecting the two words, worried and exam. So the correct statement would be, I am worried about the exams. Identify the meaning of the idiom from the option given. Hit the road, end the journey, an accident, begin the journey, or happy times. Generally, the idiom hit the road mean to leave a place to start a journey. Just like as we say, we got so late, we have to hit the road so that we can reach on time. So option C would be the correct answer. What will come in place of the question mark which is given in the expression? So here an equation is being given and we have to find out the correct answer. So if you solve this question, then if you apply the BODMAS, then we will find that the question mark, the value of question mark would be 2476, which is the option B. So option B is the correct answer. Given below are the two statements. One is labeled as assertion A and other is labeled as reason R. Assertion A is, for making statue we are using bronze. That is correct statement. That we use bronze since time immemorial for making statues, medals, or any other items. The reason which is given is, through the mixing of copper and tin we produce bronze. Definitely bronze is being made by mixing the copper and tin, but that can't be reason for making statues. We make statues by using bronze because bronze is an alloy, and alloys, they impart stability to the statues. As we know that a statue has to bear a humidity, they have to bear the air which is very much potential in doing corrosion. So just to save from the corrosion, statues are being made by using bronze. So the reason which is being given, that cannot be the correct reason for the assertion. So option B would be the correct answer, that we use bronze because it's a ductile, it's a malleable, and it also imparts the corrosion free environment. So reason would be this, not the one which is given as a reason. So option B is the correct answer. The sum of the five numbers is 260. The average of first two numbers is 30 and the average of last two number is 70. So what is the third number? If we solve this, as we know, the average of first two number is 30. So we can approximate that the sum would be 60 for first two numbers and the average of last two number is 70. So that means we can assume that the last two statement or last two words will be equal to 140. So now the total would be 140 plus 60, that is around 200. So the rest value, that means the middle one, would be close to 60. So option D would be the correct answer. 36 workers can finish a piece of work in 14 days. If the work is to be completed in 8 days, how many extra workers are required? So let's solve this question. If we see that the number of persons which are required for completing the work in 8 days is x, then we can relate that x by 36 is equal to the 14 by 8. And if we solve it for the x, then the value comes of 63. So 63 persons are basically required for doing the work in the 8 days. But we were doing it with the 36%. So now extra would be the 27% which would be required to cover the work in properly in 8 days. So option C would be the correct answer. What should come in the blank space in the following number series? 0, 2, dash, 18, 32 and 50. See, to solve these kind of a question, we have to find out what is the pattern they are following. So if we see the pattern, then we will be finding out that they are following a formula of 2 into n raised to the power 2, where n is the number starting from 0. So if we solve 2 into first 0 square, that would be the value will be 0. And if we solve it for the further values like 1, 2, 3, 4, 5, then we will find out that the pattern is being followed. So the missing word would be the 8, which is coming in the third case. So option D would be the correct answer. Three of the following four are alike in certain way based on the English alphabetical series and so form a group. Which is the one that does not belong to that group? WXST, XCDE, QRMN or IJEF. See, to solve these kind of questions, we have to find out what pattern they are following. Like in the case of A, it is ST, UV is missing, WX. In the option C, MN, OP is missing, QR. In the option D, EF, GH is missing, IJ. All three option A, option C and option D, they are following a pattern which is missing in the case of option B. So option B is the correct answer. If every alternate Saturday and all Sundays are holidays in a 30 days month beginning on Saturday, then how many working days are there in that month? Is it 21, 22, 23 or 24? If you see it in the figure, then there would be 5 Sundays would be there, that is on 2nd, 9th, 16th, 23rd and 30th. While there would be 2 Saturday which will be observed as a holiday, that is on 8th, that is 2nd Saturday and 22nd. Rest 23 days would be the working day. So option C is the correct answer. Pointing to a woman, Neetu said, her son's father is the son-in-law of my mother. How is Neetu related to the woman? Is she his mother, sister, aunt or mother-in-law? So Neetu is pointing out to a person or to a lady and she is saying her son's father is her husband. So obviously she is talking about a lady whose husband is the son-in-law of a lady which is the mother of the Neetu. So in any way it will be only possible if the lady which she is pointing is her sister. So option B would be the correct answer. Sunita traveled 5 kilometers towards east direction. She turned right and went for 3 kilometers. Then she turned west and traveled for 1 kilometer. How far she is from the starting point? So if we see the diagram, then we will find out that she started from point A and went up to B, that is 5 kilometers. Then she moved from B to C, that is for 3 kilometers, and from C to D, that is for 1 kilometer. Now we have to find out how far she is from point A. That means in a way we have to find out the distance from A to D. If we see with a point E, which is an imaginary point, then we can find out that BCDE forms a rectangle. And now the remaining distance, that is AE, would be of 4 km, while DE would be of 3 km. So in a triangle AED, if we solve it, then we will find out that the distance of AD would be 5 km, which is the option B. So option B is the correct answer. In a certain code, temporal written as a word. Now in the same way, by following the same pattern, what would be the code for consider? So we have to find out in this case, we have to find out the relation. So let's relate temporal with the word olds MBSP. So if you relate these words, then we will find out that T is related to the fourth alphabet, while E is related to the D, M is related to L, P is related to O, and O is related to P, R is related to S, A is related to B, and L is related to M. So they are the adjacent alphabets. So if we follow the same analogy for consider, then we will find out that the answer would be the option A, that is RM, NB, SF, EJ. Whose creation are Harshcharitra and Kadambari? Is it Kalana, Panini, Banabhatta or Patanjali? Basically, Banbhatta was a Sanskrit poet of Emperor Harshvardhan time, and he was the one who wrote Harshcharitra and Kadambari. So option C would be the correct answer. Present day Istanbul in Turkey was earlier known as, was it Ankara, Ottoman, Constantinople or Byzantine? Basically, if you see the map, the Istanbul, which was also known as earlier Constantinople, it was an important trade route which was connecting the Europe, Russia and Asia. and this connecting link was very much entertained in the past. So present day Istanbul is also known as earlier Constantinople. So option C is the correct answer. SpaceX is an Indian auto giant, a movie on space, name of a spy agency, or an American private space company. SpaceX, which is also known as Space Exploration Technology Corporation, was an American aerospace venture which was founded in 2002, and hence the option D would be the correct answer. UN Day is celebrated on 26th November, 24th November, 26th of October, or 24th of October. 24th of October is celebrated as UN Day since 1948, and year 2020 marked the 75th anniversary of the UN and its founding charter. So option D would be the correct answer. The famous novel Middlemarch was written by George Eliot, William Wordsworth, William Shakespeare, or Ruskin Bond. Middlemarch, a study of provincial life, is a novel which was written by an English poet, George Eliot, which was published in 1871 to 1872. So option A would be the correct answer.
चलिए, पार्ट ए के क्वेश्चंस हमने डिस्कस कर लिए हैं। अब हम एंटर करेंगे बायोलॉजिकल डोमेन के क्वेश्चन में, पार्ट बी में, जो कि 75 क्वेश्चंस हैं और अक्रॉस द बायोलॉजिकल डोमेन को कैप्चर करेंगे। तो उम्मीद करते हैं कि आपको हमारे एक्सप्लेनेशन इस डोमेन में भी पसंद आएंगे।
Which of the following electronic transition is not possible in UV visible region? N to pi star, sigma to sigma star, pi to pi star, or none of these. As we know that UV, which starts from 190 to 380 nanometer, while visible, which starts from 380 to 750 nanometer, they have got sufficient energy to promote electronic transition from ground state to excited state. Pi to pi star and N to pi star, these are the low energy required electronic transition, and they can enough be promoted by the UV visible range. While sigma to sigma star transition require high energy for electronic transition, and they are the outside the scope of UV visible range. Hence, the option B should be the correct answer. Resolving power of a microscope mainly depends on numerical aperture, refractive index, objective lens, or ocular lens. As we know that the resolving power is the ability to distinguish two closely situated points as two distinct ones. If we see the equation, that we will find that the point of resolution or the limit of resolution can be related with the lambda and numerical aperture. Generally, the resolving power is directly related to the wavelength, while inversely related to the numerical aperture. So here in the option numerical aperture is being given, so the option A would be the correct answer, that resolving power of a microscope generally depends on the numerical aperture in an inverse relationship. Which one of the following is not true according to Kasha's rule about fluorescence? Bathochromic shift is observed in emission relative to absorption. Hypsochromic shift is observed in absorption relative to emission. Bathochromic shift is observed in absorption relative to emission, or none of these. Before we understand Kasha's rule about fluorescence, we should know what is bathochromic and hypsochromic shifts. If your emitting wavelength after excitation is being shifted to higher wavelength, then it is called as red shift or bathochromic shift. While if this emitting wavelength is shifted to the lower wavelength, then it is called as hypsochromic shift or the blue shifts. Now, as a general rule, in fluorescence, when fluorophore is excited at one particular wavelength, then it goes to the excited state, and upon returning to the ground state, it definitely loses certain energy. Generally, after losing its energy, the wavelength which is being used as an emitting wavelength, that has a higher wavelength. That means what? The excitation wavelength will be shifted to the higher wavelength after emissions, and that's where it has to be the bathochromic shifts. So if you see it in the option C, bathochromic shift is observed in absorption relative to emission. That is wrong. Basically, emission is being shifted to the higher wavelength. That means bathochromic shift is observed in the emission rather than absorption. So option C is the correct answer. In reverse phase liquid chromatography, stationary phase is polar, non-polar, hydrophilic or ionic. As we know that Sweat, it invented chromatography where he used cellulose paper as an stationary phase and hexane as an mobile phase. Since hexane is a non-polar while cellulose, which is a polar, if we use such kind of a system, then we are into the normal phase chromatography. That means stationary phase is polar while mobile phase is non-polar. If we reverse the process, that means stationary phase is non-polar while mobile phase is polar, then we are using the reverse phase chromatography. So in the reverse phase liquid chromatography, stationary phase has to be non-polar, that is option B. Diethyl aminoethyl DEAE cellulose is an anion exchanger. It will prefer to bind with proteins having no charge, positively charged, negatively charged, or binding does not related to the charge. DEAE, since it is having amino group, so it is a positively charged resins which can bind to the negatively charged amino acids or protein. So option C has to be correct. Increase in the half cell potential, that is E naught, decreases the oxidizing ability, increases the reducing ability, increases the oxidizing ability, or increases the reaction spontaneity. As we know, the ability of the substance to make other substance to lose its electron is called oxidizing power. With an increase in the standard electrode potential, the oxidizing power of a substance increases. So option C has to be the correct answer. Lowest yield of ATP is achieved in fermentation, anaerobic respiration, aerobic respiration, or both A and B. Generally, aerobic respiration yields you the highest amount of ATP, that is around 36 to 38 ATP after complete electron transport chain. Anaerobic respiration, which does not involve oxygen as a final electron acceptor, rather they use sulfate, nitrate or even carbon dioxide as a final electron acceptor and may continue up to electron transport chain. And they can yield you a net gain of 2 to 36 ATP, 2 in the case if they are not continuing with the Krebs cycles and electron transport chain. Fermentation, which always stops after glycolysis and never continue till electron transport chain, can yield you a net gain of 2 ATP. Hence, the lowest yield of ATP can be observed only in fermentation. Which of the following is a molecular motor? Kinesin, kinetin, keratin or dinectin? Kinesin, dynein and myosin. These are the example of three cytoskeletal motors where kinesin moves cargo inside cells away from the nucleus along microtubules. So option A has to be the correct answer. The key driving force for membrane transport is membrane thickness, distance, molecular size or concentration gradient. That's very easy. The key driving force for the membrane transport is concentration gradient. If it is not being facilitated by a transporter, then it is an example of normal diffusion where the molecules are being transported, generally hydrophobic nature. They are transported across the plasma membrane from higher concentration gradient to the lower concentration gradient. If we are talking about some polar molecules or hydrophilic molecules which need to pass the cell membrane, then they might be facilitated by certain transporter from the highest concentration gradient to the lower concentration gradient, downhill the concentration gradient. And such kind of a diffusion is known as facilitated diffusion. So here option D has to be correct answer, that the key driving force for membrane transport is always be concentration gradient. Sedimentation rate of a particle will be zero when density of the particle is less than the surrounding medium, density of the particle is more than the surrounding medium, density of the particle and the surrounding medium are equal, density of the particle is twice as the surrounding medium. As we know, in this question, it is being asked that the rate by which a particle is being drowning will be zero. That means the particle is no longer is being sedimenting. It will be when the density of the particle and the surrounding medium will be equal, then a particle will be in a kind of a hover situation. It is no longer will be drowning. If the density of the particle is less than the surrounding medium, then it will always be floating. If the density of the particle is more than the surrounding medium, that means it is a heavier than the surrounding medium, so it will always sediment. So here the option C has to be the correct answer, that whenever the density of the particle and the surrounding medium are equal, then it will always be in a hovering condition. This is the same concept which we used in the isopycnic type of gradient centrifugation. So option C has to be the correct answer. 70S ribosomes is made up of 50S and 30S subunit, 50S and 40S subunit, 60S and 30S subunit, 60S and 40S subunit. A prokaryotic ribosomes, which is 70S, is made up of two subunits, that is large 50S subunit and a smaller 30S subunit, where each subunit consists of one or more ribosomal or rRNA molecules with specific protein. So here the option A has to be the correct answer. For each ATP molecule breakdown, the sodium potassium pump causes movement of more sodium out and less potassium in, potassium out and sodium in for 1-4-1 basis, sodium out and potassium in on a 1-4-1 basis, potassium and sodium in the same direction. If you see it in the diagram, the sodium potassium pumps moves 3 sodium out and 2 potassium in for each ATP hydrolysis. So it moves more sodium out and less potassium in, which is option A. So option A is the correct answer. The number of a nuclear pore in a cell depends on the size of the cell, transcriptional activity of the cell, DNA content of the cell, or none of these. Nuclear pore is a point of communication between cytoplasm and nuclear compartment. When transcription is necessary, then these pores exist in large number during the cell cycle. For example, in a typical mammalian cell, generally we observe that the number of these nuclear pores they gets in double during the G1 and G2 phase of a cell division. So the number of nuclear pores in a cell depends on the transcriptional activity of the cell. Option B. Which of the following is not a part of endomembrane system? Golgi body, lysosomes, ribosomes, or vesicles. Endomembrane system include nuclear membrane, ER, Golgi apparatus, lysosomes, vesicle, or endosomes. Ribosomes definitely are not the part of these endomembrane system. So option C has to be the correct answer. Michaelis-Menten constant, that is Km, is related to the enzyme's affinity for the substrate, enzyme's activity for the substrate, enzyme specificity for the substrate, or enzyme selectivity for the substrate. Km or Michaelis constant can indirectly be related to the enzyme's affinity for its substrate molecule. Generally, it has been seen that natural substrate they do have the lowest Km value for an enzyme. However, this could be misleading as well, because it has been seen that certain inhibitors, which are also known as suicidal inhibitor or tight binding inhibitors, their Km value is far less than the Km value for a natural substrate for an enzyme. So here we can say that option A is close to correct among the given option. The organelle absent in a plant cells is vacuole, lysosomes, chloroplast, or centriole. Centrioles are the organelle which are generally absent in the plant cells, and they do not use centrioles during cell division. So option D has to be the correct answer. Which of the following is not associated with the ECM of the cells? Proteoglycans, cellulose, collagen, or fibronectin? If you see in the figure, ECM is primarily composed of proteins like collagen, non-collagenous proteins like fibronectin and elastins, while a combination of carbohydrate and a proteins shaft, that is proteoglycans. On the contrary, cellulose is found in the plant cells while absent in the animal cells. So option B has to be the correct answer. The enzyme responsible for the removal of DNA supercoils is helicase, topoisomerase, DNA ligase, or DNA polymerase. Topoisomerases are the enzymes which are responsible for removing supercoils in the DNA helix, and they do it by transiently cleaving one or both the strands of a DNA. If one of the strands is being cleaved, then it will be done by the topoisomerase 1, while if both the strands are being cleaved, then it will be done by the topo 2, or also known as DNA guidase. So topoisomerases are the enzymes which are responsible for removing the DNA supercoils. Which of the following is not a secondary messenger? Calcium, diacylglycerol or DAG, cyclic AMP or AMP. Secondary messenger are the intracellular signaling molecule which are released in the cytoplasm in response to an hormone when it binds to a receptor. Some of the common examples of secondary messengers include cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol or DAG, calcium ions, or even nitric oxide molecule. The option which does not fall in the category of secondary messenger would be AMP, that is option D. Intramolecular rearrangement are catalyzed by transferases, oxidoreductases, isomerases, or lyases. Isomerases are the general class of enzyme that convert a molecule to its isomers or an alternate arrangement of the structure, which is also called as intramolecular rearrangement. So option C has to be the correct answer. An enzyme without its required cofactor or prosthetic group is referred as coenzyme, holoenzyme, apoenzyme, or catalyst. Apoenzyme is the protein part of an enzyme without its cofactor or coenzyme. So option C has to be the correct answer. Gram staining is based on the presence of dash in the cell wall, phospholipids, peptidoglycan, lipid A, or glycoprotein. Gram staining is based on the amount of peptidoglycan layer present in the bacterial cell wall. Those bacteria which has the thick peptidoglycan layer will be considered as positive after Gram staining process. So option B is the correct answer, that Gram staining is based on the presence and amount of the peptidoglycan layer in a bacterial cell wall. Lipopolysaccharide present in gram-negative bacterial cell wall is considered as an endotoxin because of: is it lipid A, is it core polysaccharide, O side chain, or integral protein? Lipid A is a lipid component of an endotoxin responsible for the toxicity of gram-negative bacteria. If you see it in the figure, then you will find that lipid A is the innermost region of the entire toxin and has hydrophobic group attached with the carbohydrate side chains. So option A is the correct answer. Antibacterial action of penicillin could be attributed to inhibition of the enzyme transpeptidase, inhibition of the cross-linking of peptidoglycan side chain, hydrolysis of peptidoglycan, or both A and B. Penicillin and other beta-lactam ring based antibiotic, they inhibit the antibacterial action of a penicillin derived from the inhibition of enzyme transpeptidase, which in turn inhibit the cross-linking of peptidoglycan side chain. So option A and B both are correct. So B has to be the correct option. Severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2, is caused by a single-stranded RNA virus, double-stranded RNA virus, single-stranded DNA virus, or double-stranded DNA virus. As we know, the SARS-CoV-2 is a single-stranded RNA virus of approximately 30 kb genome size and belongs to the type 4 category of Baltimore classification. So option A has to be the correct answer. For exergonic reaction, delta G naught is negative and KEQ is more than 1. Delta G naught is positive and KEQ is more than 1. Delta G naught is negative and KEQ is less than 1. Or delta G naught is positive and KEQ is less than 1. As we all know that the exergonic reactions are generally spontaneous in nature, which do occur by releasing free energy to their surroundings. So definitely as a logic, the product will be left with less free energy than the reactant, and overall sign of free energy change would be negative. By losing free energy, generally exergonic reaction tend to reach equilibrium, and the reactions which are having KEQ more than 1, they move spontaneously towards the product formation by releasing their free energy. So what will be the conclusion? For an exergonic reaction, the delta G naught will be negative. As you know, that product will be having less free energy than the reactant, and KEQ will be always more than 1. So option A has to be the correct answer. 2-keto 3-deoxy 6-phosphogluconate is an intermediate of pentose phosphate pathway, Antner-Dodorov pathway, Amden-Meyerhof pathway, or TCA cycle. 2-keto 3-deoxy 6-phosphogluconate is an intermediate of Antner-Dodorov pathway. So option B has to be the correct answer. Product of dissimilatory nitrate reduction is ammonia, nitrogen, nitrate, or nitric oxide. Dissimilatory nitrate reduction is in a way similar to denitrification, and in another way it is different from denitrification. If we talk about similarity, that both the reaction starts with nitrate. In the dissimilatory nitrate reduction, the nitrate is being terminally converted into ammonium ion, while in the denitrification, the nitrate is being converted into the nitrogen, which is being lost to the environment. So dissimilatory nitrate reduction is more efficient because it conserves the nitrate into the ammonium and never allow it to release into the environment. So option A is the correct answer. Which of the following is not a type of microtubule associated with mitotic spindle? Polar, astral, central, or kinetochore? If you see it in the figure, then you'll find out that the astral, polar, and kinetochore, these are the three main types of microtubule found during mitosis, while central is not a part of microtubule associated with mitotic spindle. Which one of the following is the correct sequence of prophase 1 stage? That is very simple. If you have to remember the sequence of the stages in prophase 1 stage, then you can use the mnemonics like lazy lebra, push, dipu, didi. Now, lazy stands for lactotin, zebra stands for zygotin, push stands for pectitin, while dipu stands for diplotin, and didi stands for dikinases. This is the simplest way by which you can arrange them and remember the correct sequence of prophase 1 stage. So option D has to be the correct answer. Histones are rich in lysine and arginine, lysine and asparagine, glycine and lysine, leucine and arginine. Histones are rich in basic or positively charged amino acids like lysine and arginine, so that they can bind to the negatively charged DNA backbone. So option A has to be the correct answer. Which one of the following is not true for euchromatin? They are transcriptionally active. They are less condensed region of chromosomes. Bar body is an example of euchromatin, or all of these. Euchromatin, they are the uncoiled and transcriptionally or genetically active chromatin, while heterochromatin are firmly packed and genetically inactive chromatin. So option A and option B both are correct for euchromatin. Now as far as bark body is concerned, it is a heterochromatin and tightly packed type of chromosomes, whereas functional X chromosome is covered by the euchromatin. So functional X chromosome is a euchromatin, while bar body is an example of heterochromatin. So option C is the correct answer. But here is the trick. If you see, if we say all of these, that is also not correct for the euchromatin. Think about it. Proteins responsible for structural maintenance of chromosomes are condensin, cohesin, collagen, or both A and B. Structural maintenance proteins are composed of two major proteins, cohesin and condensin. Cohesin glue up the sister chromatid together, while condensin, it helps in condensing the chromosomes. So option D has to be the correct answer, that the proteins responsible for structural maintenance of chromosomes are condensin and cohesin. Activity of protein kinase A is regulated by adenylyl cyclase, phosphodiesterase, phospholipase C, or phosphorylase kinase. Protein kinase A or pKa is a family of enzymes whose activity is dependent upon cellular levels of cyclic AMP. So option A is the correct answer because adenyl cyclase is a responsible enzyme for the converting of ATP to the cyclic AMP. So option A is the correct answer. Pectin is a polymer of glucose, glucuronic acid, galactouronic acid, or iduronate acid. Pectin is a complex polysaccharide which consists of methoxyesterified poly alpha 1,4 galactouronic acid polymer. So option C has to be the correct answer. Triple helical coil structure is a characteristic of, that is very easy. In a collagen, generally we observe that three left-handed helix, they wrapped in right-handed fashion to form a triple helix. So here the option B has to be the correct answer. Which one of the following is not true for sliding filament model? Shortening of I band and S zone with no effect on A band, shortening of I band, A band and H zone, no change in I band and H zone with shortening of A band, or shortening of I band and A band with no effect on S zone. First of all, you should know in a sarcomere what is I band, what is S zone and what is A band. If you see, when muscles are relaxed, then A band is what? The entire range of the myosin present in a sarcomere unit. H zone is what? The uncovered myosin length which is not covered with the actin. Well, what is I band? The difference between the two consecutive myosin units. So when such sarcomere unit they get contracted or muscles contraction happens, then you can see I band reduces, H zone reduces, but A band remains unchanged. So option A has to be the correct answer. Which of the following protein is associated with amphibian development? Collagen, catenin, fibrin, or keratin? If you see it in the diagram, when the fertilization happens, when the sperm fuses with the egg, then transiently a cortical rotation is observed in the amphibian egg. Now there is a asymmetric accumulation of beta catenin happens, and wherever this accumulates, that side is going to form the dorsal axis of the body pattern. So catenin is associated with the amphibian development. In fact, initial amphibian development and axis formation. Which of the following is not a pattern of cell movement during gastrulation? Ingression, epiboly, invasion, or invagination? If you see it in a diagram, then you will find that ingression, epiboly, invagination, involution, delamination, these are the pattern of cell movement during gastrulation, while invasion is not an example of the cell movement during gastrulation. So option C is the correct answer. NRSE stands for notochord restrictive silencer elements, neural ectodermal specification and answer, neural restrictive specification and answer, or neuron restrictive silencer elements. NRSE is an acronym used for neural restrictive silencer element, that is option D, which can be used as a gene switch to make it on and off of the transcription unit in any given system. In natural condition, NRSE binding protein, that is REST, is present in all cell types except neuronal cells. Once REST binds to the NRSE, it silences the neuron specific gene in non-neuronal tissue. While if REST is not present, which is the case of neuron, so neuron specific genes remain expressed. So option D is the correct answer. Which of the following is not true for master regulatory transcription factor? It must be expressed when the specification of a cell type begins. It must regulate the expression of a gene specific to that cell type, it must be able to redirect a cell fate to this cell type, or must be expressed when the specification of a cell type ends. Master transcription factors, these are the topmost regulators of gene expression, and obviously they should regulate the basal rate of transcription and can begin the transcription of a particular gene. Definitely they should be expressed before a decision need to be made about a cell lineage, rather than when cell is already differentiated. Hence, a transcription factor must be expressed when the specification of a cell type begins, rather than when the cell type's specification ends. So option D is the incorrect as far as master regulatory transcription factor is concerned. So here option D has to be the correct answer. Difference between chromatin regulation by high and low CPG promoters is: high CPG content promoters repress gene by DNA methylation. Low CPG content promoters repress gene by histone methylation. High CPG content promoters repress gene by histone methylation, or low CPG content promoters repress gene by histone acetylation. Actually, based on the CPG content, mammalian promoters could be either high CPG promoter or low CPG promoters. Cytosine in the CPG nucleotide can be methylated to form 5-methyl cytosine. Such methylation event suppresses DNA activity and may change gene expression. This activity of DNA methylation is often found in high CPG promoters, which are present in regulatory genes. These regulatory genes of having high CPG promoters, they regulate the gene expression of housekeeping gene. Generally, it has been observed that housekeeping genes have low CPG promoters. So obviously they will be having low DNA methylation as well. So having high CPG content promoters, they generally observe more DNA methylation, and thereby they regulate the activity of housekeeping genes, that is option A. Amphibian eggs undergoes equal radial holoblastic cleavage, unequal radial holoblastic cleavage, equal radial meroblastic cleavage, or unequal radial meroblastic cleavage. As we know that the amount of yolk and its distribution in an egg decide the kind of a pattern cleavage is going to follow. If suppose an atypical amphibian egg which is having less yolk content, if we follow the cleavage pattern, then we will see that cleavage actually gets completed. It divides the egg into a complete portions, and that's where has to be holoblastic cleavage. Now, if this cleavage produce asymmetric blastomers of unequal sizes as we see it in the amphibians, then this would be of unequal holoblastic type. Now, one more pattern we observe that in amphibian eggs, this unequal blastomers, they are gets arranged in a radial fashion, that means one over another. Hence, cleavage in amphibian egg would be radial, unequal, and holoblastic type, that means option B. Which substance determine the type of cleavage during development? Nucleus, cytoplasm, yolk, or A and B, that means nucleus and cytoplasm. As we have already discussed in the previous question, that the amount of yolk and its distribution pattern determines the type of cleavage. So option C has to be the correct answer. Which of the following are not true for Carl Ernest von Bayer's principles of law of embryology? A. General features of large group of animals appears earlier in development as compared to the specialized features of a smaller group. B. General features of a large group of animals appears later in development as compared to the specialized features of a smaller group. C. Early embryos of a higher animal look like a lower animal. D. Early embryo of a higher animal is look like embryo of a lower animal. Now, out of the given pair of statements, we have to find which of the following is not true as far as Bayer's law of embryology is concerned. In 1828, Carl Ernest von Baer actually proposed 4 laws of animal development. So let's first understand what he has said. In his own words, the first law was the journal character of the big group to which an embryo belongs appears earlier than the spatial character. The next law was the less journal structural relations are formed after the more journal and so on until the most specific appears. It is very difficult to comprehend these two statements. Let's simplify these. If you can recall this slide, as you know that whether it's a human, chicken, turtle, salamander or fish, the earlier embryo, they look like same. You cannot differentiate which of the following belongs to chicken while which of the following belongs to turtle. That means what? That the journal feature of a large group of animal, they appear earlier in development. In early stage of the development, you cannot differentiate. All of them belong same. All of them has a almost same character as compared to the specialized feature. Specialized feature, if you can see, that means the features where you can differentiate whether it's a turtle embryo or human embryo, that you can observe it at the later stage of development. So definitely statement A is correct, that the journal feature of a large group of animals appear earlier in development as compared to the specialized features of the smaller group. What about B? General features of a large group of animals appear later in development. That is incorrect. It appears early in the development. Now, what was his third statement? The embryo of any given form, instead of passing through the state of other definite forms, separate itself from them. And what was the fourth statement? Fundamentally, the embryo of a higher animal form never resembles the adult of another animal form. Now, again, let's simplify these two last statement about Bayer's laws of embryology. If you can see, if you have to predict which embryo belongs to human, which embryo belongs to turtle or a chicken or a salamander or a fish, you can do it at the later stage of the development rather than early stage of a development. That's very correct. Now, as far as later stage of the development is concerned, human never first become a fish or salamander or turtle rather than it becomes human. So that one thing is very clear, that human follows its own course of development. It never first become a fish or chicken or any other evolutionary lower forms. It always forms its own course of development, and that's where the early embryo of a higher animal never look like a lower animal. It only look like that its own version of adult at the later stage of development. So as far as statement C is concerned, that early embryo of a higher animal is look like a lower animal. No, it never look like an animal, lower animal. It definitely is a miniaturized version of its own adult. Option D. Early embryo of a higher animal is look like an embryo of a lower animal. Yes, at the early stage of the development, all the embryo they look like same. So whether it's a human embryo or a fish embryo, they look like a same. So early embryo of a higher animal, it is as same as like a early embryo of a lower animal. So which two statements are incorrect? Statement B and statement C. So here the option C has to be the correct answer. Enzymes involved in RNA interference are dicer, coenzyme, bicoid, or masking. As we know, the dicer is an enzyme which plays a role in the initiation of RNA silencing by recognizing double-stranded RNA and cleaving into small RNAs using its RNase 3-like activities. And thereby, these dicer regulate the biogenesis of Mi or siRNAs. So, option A has to be the correct answer. CRISPR is. So there are 4 full forms are given and we have to find out which one represents CRISPR. So CRISPR actually stands for clustered, regularly interspersed, short palindromic repeats, and it is an important gene editing tool as a method of precise DNA editing for which Jennifer Doudna and Emmanuel Charpentier got Nobel Prize. So option A has to be the correct answer. Ramachandran plot is used for the determination of stable protein structure, also called as phi-psi plot, provide information about statistically favorable conformations, or all of these. Ramachandran plot, or also known as phi-psi plot, it's a two-dimensional graph which is used to predict statistically favorable or restricted conformations for a given amino acid sequence. And thereby, we may make a guess that what is the stable conformation which could be naturally present for a given sequence. So, Ramachandran plot is definitely used for the determination of a stable protein structure. It is also called as phi-psi plot, and it also provides information about statistically favorable conformation for a given amino acid sequence. So option D has to be the correct answer, all of these. Dextrans are polymeric glucose, polymeric galactose, polymeric fructose, or polymeric lactose. Dextrans are the glucose polymer having poly alpha D glucosides of microbial origin. So option A has to be the correct answer. According to Bohr effect, the oxygen transportability of hemoglobin is more when pH decreases, pH increases, pH remains stable, or it is not affected by the pH. According to Bohr effect, the low pH and high carbon dioxide concentration ensure that more oxygen unloading is being performed from hemoglobin. Actually, in the presence of high carbon dioxide, hemoglobin shows preferential carbon dioxide binding rather than oxygen, and hence oxygen actually gets delivered at the site. Now, as far as decreased pH is concerned, that means at low pH, the reversible binding of oxygen by replacing carbon dioxide is prevented. So both decreased pH and high carbon dioxide actually cause the oxygen unloading from hemoglobin. So option A is the correct answer. Cells are small in size because it provides more surface area to volume ratio. It provides less surface area to volume ratio, provides regulation of cytoplasm by the nucleus, or option A and B both. Actually, cells generally remain small in size to enable different regions of the cells to communicate with each other rapidly and maximize surface area to volume ratio for better diffusion of the substances. So in this case, cells are small in size because it provides more surface area to volume ratio. But always remember, there is another reason for being smaller in size as far as cells are concerned, so that they can well communicate with other cells. Protein purification in 2D gel electrophoresis is based on molecular weight, charge, number of subunits, or both A and B. In a 2D gel electrophoresis, in first dimension, based on the pi, or in simple words we can say, based on the charge at different pH, we separate the proteins. And now, by placing at 90-degree angle of these separated proteins, now we place them and separate them based on the molecular weight. So both molecular weight and charge is a criteria for protein purification in a 2D gel electrophoresis. DASH enzyme catalyzes conversion of fructose to fructose 6-phosphate, and it requires DASH as a cofactor. So is it phosphofructose kinase 1 or PFK1 magnesium ion, PFK2 or magnesium ion, PFK1 magnesium ion, or PFK2 magnesium ion? Actually here none of the answer is correct because the given reaction is catalyzed by fructokinase rather than PFK or phosphofructokinase. The reaction which catalyzed by phosphofructokinase is that the fructose 6-phosphate to fructose 1,6 biphosphate. So here none of the option is correct. Which of the following is not a possible catabolic fate of pyruvate? Lactic acid fermentation, citric acid cycle, ethanol fermentation, or glycolysis. Actually, pyruvate can enter into lactate or alcoholic fermentation during anaerobic condition, while into TCA cycle during aerobic condition. Actually, pyruvate is the end product of glycolysis rather than glycolysis is the fate of pyruvate. So option D is the correct answer. Allosterically regulated enzymes are oligomeric proteins, possess regulatory and catalytic domains, show sigmoidal behavior, or both option B and C. Actually, all allosteric enzymes are multi-subunit or oligomeric in nature. But all oligomeric protein need to be allosteric? That is not correct. They do have two types of sites, one where actually activity occurs and other which are the regulatory sites where modulator binds and regulate the main activity of enzyme. Remember, one of the major assumptions of Michaelis-Menten kinetics was that protein must not be allosteric. Hence, all allosteric enzyme shows sigmoidal curve rather than classical hyperbolic curve as observed for Michaelis-Menten kinetics. So, option D, that is statement B and statement C, is correct for the allosteric enzyme. Which of the following regulation occurs in slowest time frame? Feedback regulation, allosteric regulation, regulation through covalent modification, or enzyme synthesis through gene induction. Feedback regulation, covalent modification, and allosteric regulation. These are the three rapid methods of metabolic regulation, while enzyme synthesis through gene induction is a slowest mechanism which affects the biometabolic processes. So option D is the correct answer. The unit of maximum growth rate is hours inverse, hours, hours per liter, or hours per cell. Maximum growth rate or mu max is an important parameter which is present in Monod equation. Monod equation is an adaptation of Michaelis-Menten equation for modeling microbial growth under batch condition, where mu has a unit of time inverse, that means hours inverse. So option A is the correct answer. Mammals do not have ability to convert fatty acid to glucose because they cannot undergo glyoxylate cycle. There is no need for such conversion. They can obtain glucose from other pathways or both option A and C. Glyoxylate cycle has a potential to convert acetyl-CoA, which might be coming from fatty acids, into the glucose. This cycle is found in plants, bacteria, protistans, and fungi, and definitely absent in humans. Humans lack two important enzymes of glyoxylate cycle, that is isocitrate lyase and malate synthase, which can convert fatty acid to glucose. Hence, option A is the correct answer. Formation of glycogen cannot be initiated in the absence of enzyme glucokinase, glycogenin, glycogen phosphorylase, or glycogen kinase. Glycogen synthesis cannot be initiated without glycogenin protein, onto which the activated glucose molecules are tethered sequentially by the enzyme glycogen synthase. So here the option B is the correct answer. Glucose sensors utilizes dash as a bioreceptor. Glucose oxidase, glucose kinase, glucose dehydrogenase, or both A and C. Common enzymes which are used in making glucose sensor for glucose detection uses glucose oxidases, NAD dependent glucose dehydrogenases, FAD dependent glucose dehydrogenases or glucose dehydrogenases PQQ. Out of all types of glucose related enzymes, FAD dependent glucose dehydrogenase is the most employed enzyme in glucose sensing industries. So option D has to be the correct answer, that both glucose oxidase and glucose dehydrogenases of various types can be used to construct a glucose sensor. Optical density is based on light refraction, light absorption, light diffraction, or light scattering. Optical density is a technique for evaluating the amount of light scattered by the particles suspended in a liquid sample. So option D is the correct answer, that optical density is based on the light scattering phenomena. Purity of DNA samples can be checked by measuring the ratio of absorbance at 260 and 280 nanometer. For a pure DNA sample, the ratio should be 1.8, less than 1.8, less than 1, or greater than 2. The 260 by 280 ratio of around 1.8 is generally accepted as a signature of pure form of a DNA. While if this ratio is around 2, then we can be assured that the sample contains purely RNA. If the value is quite less in favor of A260, that means that nucleic acid sample has a severe protein contamination. So here the option A is the correct answer, that a ratio of 1.8 of 260 by 280 is a characteristic that the DNA sample is pure. ATP molecules produced by the oxidation of one molecule of palmitoyl CoA to carbon dioxide and H2O are 38, 108, 148 or 40. Actually, palmitic acid is a C16 fatty acid, and after 7 rounds of beta oxidation, it will produce us the 108 ATP. As we know that before beta oxidation, there are 2 ATPs are used for the activation of 3 fatty acids. So hence the net gain would be 106 ATP, but the total ATP production would be 108. So option B is the correct answer. Carbamyl phosphate synthetase 1 or CPS1 is the first enzyme in urea cycle, and it is allosterically regulated by N-acetyl glutamate, N-acetyl glucosamine, N-acetyl galactosamine, or N-acetyl gluconate. CPS1 is a hepatic mitochondrial enzyme involved in the condensation of ammonia and carbon dioxide during urea cycle. This enzyme allosterically regulated by a chemical called N-acetyl glutamate. N-acetyl glutamate is synthesized by the condensation of acetyl-CoA and arginine, which is produced during the urea cycle. Now, higher level of arginine simply means that more N-acetyl glutamate will be produced, and hence there will be more check on the CPS activity. So, option A is the correct answer. Phenylketonuria is caused by dysfunction of phenylalanine hydroxylase, phenylalanine monooxygenase, phenylalanine mutase, or phenylalanine dioxygenase. Phenylketonuria, or also known as PKU, it is caused by the genetic deficiency of phenylalanine hydroxylase, which led to the impaired phenylalanine metabolism. So option A is the correct answer. Key protein involved in complement activation is C5, C8, C9, or C3. The central event in complement activation is the proteolysis of the complement protein C3 to generate biologically active products and the subsequent covalent attachment of a product of C3 called C3b to microbial cell surfaces or to antibodies bound to antigen. So the key protein involved in complement activation, whether it is coming from classical pathway or mannose binding ligand or alternative pathway, the central protein would be C3. So option D is the correct answer. Separation of components in gas chromatography is based on molecular weight, partition coefficient, molarity, or conductivity. The principle of gas chromatography is based on partition coefficient. The mixture of components to be separated is converted to vapors and mixed with the carrier gas or mobile phase. When these vapors interact with the stationary phase, they get partitioned between mobile and stationary phase as per their chemical nature. So option B is the correct answer. Pervaporation is ion removal, separation of liquid, separation of solid, or water purification. Pervaporation is a membrane separation process in which components from a liquid mixture are selectively transported through a microporous membrane. So option B is the correct answer, that pervaporation is a method of separation of liquid. Obligate anaerobes, that means strictly anaerobes, are not able to survive in the presence of oxygen because of absence of both catalase and superoxide dismutase, presence of both catalase and superoxide dismutase, presence of catalase but absence of superoxide dismutase, or presence of low levels of both catalase and superoxide dismutase. Obligate anaerobe, they are the microbes which do not require oxygen to live, and they would die in the presence of oxygen. They lack enzyme SOD, which is superoxide dismutase, and catalase, which are used for the conversion of lethal superoxide formed in the cell when oxygen is present. So obligate anaerobes are not able to survive in the presence of oxygen because they lack catalase and superoxide dismutase. Which of the following sentences is not true for pyrosequencing? It is based on detection of pulses of chemiluminescence, that is correct. Requires the activity of enzyme sulfurylase, that is also correct. Requires addition of dideoxynucleotide, that is incorrect. If you see it in the figure, we both require the pulses of chemiluminescence and also enzyme sulfurylase in order to perform pyrosequencing. But we never use dideoxynucleotides because dideoxynucleotide is a method or a step which is used in Senger's method of sequencing rather than pyrosequencing. Incorporation of any dideoxynucleotide may further inhibit the action of DNA polymerase, as there is an absence of free 3' OH in dideoxynucleotide. So option C is the correct answer. Match the list 1 item with list 2 items. Translation, replication, ATP synthesis, and dark reaction of photosynthesis. Where do they occur? That's very simple. Translation, as we know, it occurs in the cytoplasm. Replication definitely occurs in nucleus because genome is present over there only, central genome. ATP synthesis, it occurs in mitochondria. That's where mitochondria is often regarded as powerhouse of the cell. And dark reaction, that means Calvin cycle of photosynthesis, it occurs in stroma of chloroplast. So if we collect, then option D is the correct answer, that translation which is occurring in the cytoplasm A24, replication which is occurring in nucleus B21, ATP synthesis which is occurring in the mitochondria C22, and D has to be connected with 3, that is option D. Impermeability of mitochondrial inner membrane to most ions, electrons, and protons is due to the presence of cardiolipin, collagen, chitin, or cellulose. As we know that the inner mitochondrial membrane is largely impermeable to charged ion due to the special lipid molecule constituting it. Whereas the normal phospholipids are having only 2 acyl chains, while cardiolipins has 4 hydrophobic acyl chains. And that's where, due to the increased hydrophobicity, it is very difficult for a charged ion to make it through the plasma membrane. of a mitochondria. Hence, it is the cardiolipin which ensure the impermeability of mitochondrial inner membrane to most of the charged species. Option A is the correct answer. Lateral root primordia are developed from pericycle, endodermis, ectodermis, or xylem. Lateral root primordia arises by the localized cell division in the pericycle, which resulted in the moon of the tissue. So, option A is the correct answer. Agrobacterium tumefaciens is used for the transformation in plant tissue culture because both agrobacterium and plants have similar genetic makeup. That is incorrect. It cannot be correct by any means because agrobacterium is a prokaryote and plants is a higher eukaryote. We cannot expect it to have the same genetic makeup. Option B, agrobacterium genomic DNA transfer its genetic material to the plants. No, it is also incorrect. Agrobacterium actually transfer a plasmid, that is Ti plasmid, not even full complete Ti plasmid. It's a part of the Ti plasmid which is being transferred to the plant genome. And upon integration, this tDNA, it produces a special chemical called opine for its nutrition. Actually, the transfer of this tDNA is facilitated by the expression of Vir operon gene. So, option C is the correct answer, that the Vir genes of agrobacterium aid in the tDNA transfer to the plant. Mutation in the class A gene in ABCD model of flower development leads to which pattern of floral organization? Arrangement of flower organ in a pattern in Arabidopsis plant is regulated by a set of homeotic genes like A, B, and C. When any of these genes is mutated, the original or wild flower pattern gets disturbed and changed. So let's understand what is this change is all about. If we see the wild flower, the arrangement of the whorl will be sepals, petals, stamens, and carpels going inward when both A, B, and C homeotic genes are expressed. If suppose A gene is mutated as asked in the question, then the original whorl will change to the carpel, stamen, and carpels. That is option C. Now, if B gene is mutated, then original whorl will change to sepals, carpels, and carpels. While if the C gene is mutated, then the whorl will change to sepals, petals, and sepals. So here the option C has to be the correct answer, that when the A homeotic gene is mutated, then the original whorl which has sepal, petal, stamen, carpels change to carpel, stamen, carpels. With this, we conclude this video. Please do subscribe us in large numbers if you like our content. Like, share, and comment on the video. Thank you very much.